Welcome back to the channel. Today we will be looking at the main topic, genetics. Before we get into the lesson, let us look at the objectives of the lesson. Alright, objectives of the lesson are: number 1, to define the term genetics. Number 2, to identify from the cell where the genetic materials can be found. Number 3, to name and identify the parts of a chromosome. Number 4, to define the terms that are used to describe the gene, for example, dominant, recessive, homozygous, heterozygous, diploid, haploid. And number 5, to identify the punnett square and carry out genetic crosses. Okay, we have three main terms that we are going to define, including the definition of genetics. Alright, the first term is trait. Now, a trait is any characteristics that can be passed from parent to offspring. For example, hair color, height, eye color, flower height, color of the flowers, etc. So, what this is speaking about is that we are looking at the traits of both animals and plants. Alright, so gene or traits are found in all living things. Alright, the next term is heredity. The passing of traits from parent to offspring. Now that is self-explanatory. Genetics. Now genetics is the study of heredity. So what that is saying is that genetics studied the passing of characteristics from the parent to the offsprings, whether it be animals or plants. Alright, the cell. Now, the cell is where the genetic materials are stored, but there are specific parts of the cell where the genetic materials are stored. So let us get in depth with the information. The basic unit of structure of all organisms is the cell. Some organisms, such as bacteria, are unicellular and consist of only one cell. Most organisms are multicellular and consist of large numbers of organized cells into tissues and organs. Genetic materials are found in the nucleus of the cell. Alright, so to be specific, the main point is that the genetic material, the traits of plants and animals, the characteristics that are passed on from parents to offspring, they are stored in the nucleus of the cell. Alright, let us look at some diagrams of the cell. Alright, so as you can see here, this is a diagram of the animal cell, and as you know, the structure here is the nucleus, which is referred to as the brain of the cell. You will not be able to see these chromosomes or genes only when the cell is dividing. Alright, I'll do a separate lesson on cell division, so remember to follow the channel, subscribe, and like the videos so you can get those videos when I release them. Alright, so the animal cell, it doesn't have any specific shape. So here we have the cell membrane, and animal cell, it has a small vacuole, and also it has mitochondria. There are other parts, but for this lesson we're just going to focus on the nucleus. Let's look at the plant cell. Alright, so this is the plant cell, and the plant cell has a fixed shape because of the thick cell wall that surrounds it. Now here we have the nucleus. So plant cells, they do have nucleus. They also have mitochondria similar to the animal cell. They have cell membrane. They have a vacuole. However, unlike the animal cell, the vacuole of the plant cell is much larger. Alright, it also contains a cell wall that the animal cell does not contain, and also it contains a chloroplast that the animal cell does not contain. The chloroplast stores the chlorophyll. Alright, so let's get into the structure of the chromosome. And of course, the chromosome is referred to as the chromosome, it contains the DNA, and DNA stands for deoxyribonucleic acid. Alright, All right, so let's read the information here of the structure. The chromosome, DNA, are located in the nucleus and they are only visible during cell division. Number 2, each chromosome consists of a pair of identical chromatids. All right, number 3, each chromatid is joined by a centromere. And 4, the gene is located on the chromosome. For example, gene for hair color, gene for skin color, flowers color, plant height, disease resistance, etc. All right, now this is diagram of the chromosome. Now the chromosome, as it is said, it has two chromatids, so it looks like an X. So this is one chromatid and this is another. You can look at it like this. This is one and this is another. Now the chromosomes, they are identical. So this chromatid is identical to chromatid, and what that means is that the genes that are found here and the location where they are found is similar to those that are found over here. Alright, so a chromosome normally contains identical chromatid, and what we mean is that all the gene on one is found on the gene of the other at the same position. Alright, so the gene, the exemplar's gene, it will have a lot of genes, but so one gene here is the same as the one over here. Alright, so for each gene, there are two copies of a gene on one chromosome. Remember that. And the centromere joins the chromatids together. All of that is referred to as your chromosome. Alright, so let's get into how you would write your genes. Now, as we know, a gene has two pair of a chromosome. So a gene, they comes in pair. Alright, so when you're writing a gene, you're going to write them as a pair. So let's look at what we have on the slide. Genes combination representing height can be written as. So of course, say you're writing the gene for height, or the trait height. Maybe how tall the plant is or how tall the animal is, etc. You just select a letter, right? So you can select any letter to represent them, right? Now there are three ways you can write them. One, you can use two capital letters, and that represent that the gene is dominant and it is homozygous. So two capital letters mean that this gene has two dominant. They are dominant. Both of them are dominant, and I soon explain dominant means, what dominant means. Number 2, this is an hybrid. Means it consists of one dominant and one recessive gene. It's a dominant and recessive pair. You refer to that as an hybrid. And the third way is that you have two common letters, which means that it is purebred recessive. Alright, so since we speak about dominant and recessive, let me define what they are. So the dominant gene means that these will be expressed in the animal or the plant. So let's use human being for example. Two parents have a child. Now the female has brown eye color and the male has black eye color, but the child comes with black eye color. But of course, the brown eye color is in the blood of the child, but the child does not express the brown eye color. The child expresses the black eye color. So what that means is that possibly is that the gene that is dominant would be the black eye color, because that is the one that is expressed in the child. So all the genes that are expressed when you put them with another gene over and over is referred to as a dominant. They are dominant. Those that are recessive might be in the blood of the child, but it is not expressed in the child. You can't see it when you look on them physically. It's not expressed in your physical feature. You can't see it, but it's there in the blood. Alright, so that's what it means by dominant and recessive. So the recessive one means it will not be expressed when it is put with a dominant gene. Alright, so let us get into terms describing a gene. The first one is allele. And of course, when you're doing CSEC and so forth, exam, they might ask you these common questions, alright? To either list the alleles, define them, etc. They can ask you those. If there's a genetic question, possibly might ask that. So please pay attention. All right, so the alleles, there are two forms of a gene, the dominant and the recessive, and I just explained one. All right, the dominant gene is stronger of the two genes expressed in the hybrid, representing by a capital letter. So dominant genes, you normally write them in capital letter, and when you put it with a recessive, it will normally be expressed over the recessive gene. All right, it will normally be expressed in the individual or the plant or the animal, etc. The recessive gene that show up less often in a cross, represented by a lowercase letter. All right, so you use common letters to represent the recessive gene. So remember, the gene comes in pair. They are either dominant or recessive, homozygous or heterozygous, etc. All right, so there are also two important terms that we need to define. You have genotype and phenotype. Now, the genotype, the gene combination for a trait or characteristic. So, for example, the flower color. Right, so say we're looking at flower color. The genotype would be the genes that represent that flower color. Right, it is not expressed in the physical feature. It's just the gene makeup. So you know, gene comes in pair. So it could mean that the gene is homozygous, right? Meaning it is made up of two dominant gene allele, or it is made up of a dominant and a recessive allele, or two recessive alleles. So that's what it means by genotype. So when you're asked about the genotype, you're supposed to either write them accordingly. All right, whether it is homozygous or heterozygous, etc. The phenotype, these are the physical feature resulting from the genotype. So when you're asked about the phenotype, it means the physical feature of the gene, the physical appearance of the gene. It means what trait the individual will express in the physical world. Whether the animal will be brown, black, or the flower will be red or purple. Alright, so one is the gene makeup, which is the genotype, and phenotype means the physical feature that is expressed. As you can see, the example down here, red, white, etc. Alright, continuation with the terms. We have diploid cell and haploid cell. Now, a diploid cell is the total number of chromosomes in an organism. Alright, human has 46, which is 23 pairs. Goat has 54, which is 27 pairs. Red pea has 14 chromosomes, which is 7 pairs. So, a normal cell, the maximum amount or the total amount of chromosome in a cell, we refer to that as diploid. Now, what you need to note is that each type of organism or different type of organism, they have different amount of chromosome. So that is why you're not able to cross a goat with a pig, because the chromosomes are different, right? The amount of chromosomes, they are different and so forth, right? All right, the haploid cells, this is normally is half the number of the total number of chromosomes. These are normally produced in the sex cells, sperm and egg when you're dealing with animal, or it could be pollen or ovule when you're dealing with plants. The normal amount will be restored during fertilization, the joining of the egg and the sperm. So haploid cells, the sex cells, are normally haploid. Alright, so normal body cells, normal regular cells of the plants would be diploid, but the haploid would be in the sex cells, whether the pollen grain or ovule of the plant or the sperm and egg of the animal. For instance, if the normal amount for human is 46, the haploid for this would be 23 chromosomes. So the egg would have 23, the sperm would have 23 chromosomes. All right, we have two other terms that we look at, and then we'll go into the genetic crosses. We're going to look at the punnett square and how the crosses can be done. All right, so we have two terms that describes the genotypes, and as I said, the gene makeup. So we have the homozygous genotype, gene combination involving two dominant or two recessive genes. So example, we have two capital R, two common R. And this is also referred to as pure, or I could say purebred. Alright, the heterozygous genotype, gene combination of one dominant and one recessive allele. example, capital and a common R, also called hybrid. Alright, so let us get into the genetic crosses. So we have two types of genetic crosses. We have the monohybrid cross and the dihybrid cross. Now the monohybrid cross, as it says, mono means one. So what we're speaking about is a trait. So this cross is only going to focus on one trait at a time. Dihybrid is referring to two traits. Alright. However, for CXC, we're going to mainly focus on the monohybrid. But let's do the definition for both. Monohybrid cross, this is cross involving a single trait, example, the flower color. So when we're working out these crosses, we're just going to do it for one trait. The dihybrid cross, it cross involving two traits, for example, flower color and plant height. Now we're not going to look at that one. Alright, with the important note is that with the monohybrid cross, you have 4 possibilities of offspring being formed, and with the dihybrid you have 16 possibilities of offsprings being formed. Now I'm going to explain the reason or the purpose of the genetic crosses, so please listen or please stay tuned. Alright, the monohybrid cross. So of course when you're carrying out this cross, you have one trait. Sex cells or gametes are the cells involved, not the regular body cells. So when you're doing the monohybrid cross, we're going to work out the sex cells. We're going to cross the sex cells in the punnett square. Alright, so whether it be a sperm or egg or pollen with an ovule. A punnett square is used to predict the possible gene makeup of the offsprings. Very important. So that is the main aim of a cross. Why we carry out these genetic crosses is because we want to see what traits specific offsprings will have. And let me give an example. So if you're operating a dairy farm, of course you want animals that produce large amount of milk. Say one of the animals, they produce large amount of milk that you prefer, but you have another on the farm that is not producing enough milk. However, you want offspring that will produce more milk. What you have to do is you could cross the animal, maybe a male from the animal that produce large amount of milk, with one that don't produce, and based on those data now, you could use that to say, if I cross this animal that produces a lot of milk with this animal that don't, what will my offspring be? How many percent chance I have for the offspring come with high milk production abilities? So that's what your crosses will do. It help you to make prediction. Alright, as we go in depth, you will understand more. So the other point is that our features of the cross, the monohybrid, is that 4 possible gene makeup of the offsprings are expected. Alright, so to do their monohybrid cross, or the crosses, we normally do that in a punnett square. Alright, and as stated here, is that the punnett square is used to solve genetic problems, which means to make prediction for us. Alright. Alright, the punnett square for the dihybrid cross would have 16 squares. Alright, but we're not going to focus on that. Alright, in order to help us understand how to use the punnett square, here's a CSEC past paper question. A farmer decided to improve milk production in his dairy herd. He mated 2 animals, each with the hybrid gene for milk production. The heterozygous gene, this is an heterozygous gene for milk production. So both the animals have heterozygous genes. And how they decide that, as I said, they would have to be carrying out experiments over a period of time or years to know the gene makeup of the animals. Alright, so now that they know, they want to see what will happen when they mate these 2 animals with the same gene makeup. Both animals have same gene makeup, so they get a male and a female, right? Now let's continue. Number 1, 1A. Complete table 2 to show the genotype of the F1 generation. Alright, the F1 generation means this first set of offspring from those pairing. The first offspring from those pairing would be the F1 generation. If you have those offspring now have produce offspring, that would be F2. Alright, so the F1 generation means the first offsprings. Alright, so this is the table. So here At the top, you have, say, these are the gene makeup, say, from the female. You can put the female at the top, gene makeup from the male at the side, or vice versa. You'll get the same result here in these 4 squares. So you would fill out the squares, the 4 squares with the gene. So let's go back. So this m will be matched or multiply or added. We add both of them together, you multiply them, and you'll get 2 capital M right here. So this one will be matched with this one to get this square. To get this square, you match this one with this one to get this. To get this, you'll match this m with this m. Alright, and that is the principle used for all of them. So let us go back. So if you follow the arrow here, it says that this red m here with this green here to get this square here, right? So here you have 2 capital. Let's look at this one now. Fill out this one. To fill out this one, you'll match this capital, this green m, with this common m here to get this gene makeup. And for this square, you'll match this gene with this gene here to get this. To get this gene makeup here, you match this common red m with the green one, green common m here to get this one. Alright, so what we actually do is to segregate the gene makeup and join them back. Now this process normally happens during fertilization. So when the egg meet the sperm, you're going to have fertilization, and then the new genes will be created in the offspring. So using this, you can see where you have the types of genes that the offspring will have. So there's a possibility that the offspring might come with this gene makeup, which is a homozygous dominant gene makeup. This one here is that an heterozygous, right? We have another one with the same gene heterozygous, and another one now that has homozygous recessive gene makeup. Now what I did was to put percentage. So each square represents 25%. So let us read this just for you to understand better. So animals coming with homozygous dominant gene makeup would be 25%, because it's only one square that has this homozygous dominant gene makeup, right? With this gene, we have it in this square and we have it in this square. So that's 2 squares, so that's 50%. So 50% chance of the offspring will be coming having heterozygous gene makeup, and that would be 25% chance of the offspring having the homozygous recessive gene makeup there. Alright, let's move to 1b of the question. Identify the percentage of cows in the F1 generation that will be homozygous for milk production and heterozygous for milk production. So you're going to write out the homozygous genes and the heterozygous genes from the cross. So let's look back. So this will be homozygous, this will be homozygous, this is 25, this is 25, so that's 50. This will be heterozygous, this will be heterozygous, so both of them would be 50, which is 2 out of 4 for this, 2 out of 4, which is 50. Alright, let's look how we write them out. So A, the homozygous dominant and the homozygous recessive. Based on the result, there are 2 homozygous cows out of 4 possibilities. Hence, the percentage for the homozygous cow is 2 out of 4 times 100, which is 50% is the answer. And if you look back, you can see that the homologous would be 50. 25 here, 25 here. Alright, for B, the heterozygous genes are MN, heterozygous, the dominant and the recessive. Based on the result, there are 2 heterozygous cows out of 4. Hence, the percentage for the heterozygous cow is 2 out of 4 times 100, which will give you 50% of the gene would be heterozygous. Alright, so let's look at this question. So this is the last question, guys. So let's look at this one. A doe rabbit with gene for a large candling size is homozygous dominant, and candling mean the amount of kittens that the rabbits can produce. So when rabbits give birth, we call that candling. So that is homozygous dominant is mated with buck with gene makeup is heterozygous. Alright, so buck is male, doe is female in rabbit. Alright, the larger litter size is dominant to the smaller litter size. So the capital letter would represent those this gene, the large candling size, based on experiment, would be the dominant gene, and the small candling size would be recessive. All right, 2A, complete table tree to the genotype to find the genotype of the offsprings. 2B, calculate the percentage of each genotypes and phenotypes, physical features the offspring will express. All right, so this is the table. So here you put the female gene on the up here at the top, and vertical you're going to put the male, and you can do it vice versa. All right, so when you match them up, so to do this one you match this with this, to work out this you match this one with this one, to work out this you match this with this, and to work out this one you match this with this. All right, also you need to know you put the capital letters first. All right, remember that you put the capital, so you rearrange them and put the capital first each time. All right, so according to number one question, so that's the number one answer. That's 2A answer. Right, 2B, calculate the percentage of each genotype and phenotype. All right, so that's number 2A. All right, 2B, A, the genotypes. So we're going to write out the genotypes. So it is homozygous. So let's look at the genotypes. So we have homozygous dominant here. We have the same homozygous dominant here. Right? So that's one. Two of them make up one. It's the same gene makeup, right? But this is telling you that this is a 50% because only two of the squares. Alright, the other different type would be heterozygous here, and we have the same. So these two will come together. It would be the same. So it's one. So you have one here and one there. Alright, anyways, let's see how we'd write them out. So we'd have one here, the homozygous here, and one here, which is the same. So that's why it is placed in bracket here. However, to work out the percentage, you realize that 2 out of the 4 squares, and go back to the punnett square, 2 out of the 4 squares was homozygous dominant. So 2 out of 4 times 100 would be 50%. The same thing for the heterozygous. 2 out of 4 would be 50%. Alright, so this is how you write the genes. Remember, guys, girls. And the phenotypes now. In order to decide the phenotype, then you have to know the gene. But remember that the phenotype speaks about the physical characteristic that is expressed. Alright, so this 50% homozygous dominant would mean 50% of the organism of the offsprings will be large size producer, or large candling producers, or large size producers. They produce a lot of kitten. 50% would be heterozygous, which mean large size producer as well. Even though it's homozygous, once the gene makeup has a capital letter in it, it means that the animal will express the dominant gene, which is the large size producer too. So this gene will be large size producer, this one would be large size producer. So the answer would be all will be large size producer. However, let's go back to question 1. I want to show you, make a comparison. With these two gene makeup, when you cross animals with heterozygous and heterozygous, these are the possible gene makeup you will get. But when you cross animals with homozygous dominant and heterozygous dominant, this is the gene makeup you normally get. And you might see questions where you cross homozygous dominant with homozygous recessive. You'll get a different gene makeup. Alright, this is the end of the lesson, guys. Please stay tuned, practice questions. You can make comment, you can ask questions, you can message me, you can ask me to do other videos to explain. If you have questions, you can send them to me and I'll answer them for you and get back to you. Until next time.